So today I want to talk about how we used dSTORM to visualize the regulation of neuronal GABA-A receptors spatial patterning with GABA and astrocytic cholesterol. So this is our recent publication on communicators biology, and the QR code here is to the paper. So if you're interested, you could scan and read the paper. And in today's talk, I will just mainly talk about results from this publication. So GABA-A receptor is a major anesthetic channel that mediates rapid synaptic inhibition, with the channel activation increasing chloride influx in modulating neuronal excitability. And multiple binding sites for the anesthetics have been identified with the structural studies before, but this does not exclude anesthetics' potential interaction with the membrane. And especially when we examine the chemical structure of these known ligands, we see that this definition is incredibly broad, ranging from single atoms to more complicated structures. And the only common characteristic seem to be hydrophobicity, with no clear chemical specificity. And this leads us to the lipid theory of anesthesia, which shifts the focus to the lipid environment in which the ion channels reside, and for the GABA-A receptor. And over a century ago, Mayer and Overton has this observation of a strong correlation between the hydrophobicity of anesthetic compounds and their potency, now known as the Mayer-Overton correlation. And this is a strong support that the lipid environment could be a potential site of actions for the anesthetics who are activating GABA-A receptors, but actually interacting with its local environment. And when I first joined the lab, the Hansen lab has just published this paper on the membrane-mediated mechanism for TRK1 channel in general anesthesia. And for the first time in the past 2 decades, the focus of anesthesia shifted from the channel itself to its local environment on the membrane. And to get a better idea, let's zoom into the molecular level of the membrane. Here I want to introduce the concept of spatial biochemistry as the study of how proteins are positioned within the plasma membrane and how their behavior is influenced by the surrounding membrane environment. And this positioning is not random. It's determined by the unique physical and chemical property of the membrane. As all of us know, the plasma membrane is highly heterogeneous, with lipids differing in properties like saturation levels and azo chain lengths. And this variation leads to the formation of specialized clusters, such as lipid rafts and peptide domains, as shown here. So lipid rafts are ordered microdomains enriched with cholesterol and saturated lipids, while the unsaturated peptide lipids cluster the positively charged proteins within the disorder region. And on the proteins, there are multiple translational modifications that could act as molecular tag for different microdomains on the membrane. As we can see here, the palmitoylation is more in favor of the order domain within the lipid rafts, and prenylation is another modification that's more in favor of the disorder region. And interestingly, about 1/3 of all lipid raft proteins are palmitoylated. So this suggests that palmitylation could be an important mechanism for locating these proteins within the membrane. And in our case of the TRK1 that was published in the lab, it also has binding sites for the peptide domain, suggesting these receptors would have affinity for different regions on the membrane, and there's a potential trafficking between these different domains in changing their functionality. So to explore whether this mechanism could extend beyond a single potassium channel as TRK1, we turned our attention to another palmitoylated protein as the GABA-A receptor. So we are particularly interested in the lipid environment for GABA-A receptor. We learned that from the literature, it shows that the GABA2 subunit of GABA-A receptor undergoes palmitylation, which is essential for its proper membrane expression. and this has been demonstrated using a palmitoylation inhibitor and significantly diminish the receptor's expression. And additionally, the localization of GABA-A receptor is highly dependent on cholesterol level, aligning with our understanding of cholesterol's role in stabilizing the lipid rafts. When cholesterol is removed using methyl beta cyclodextrin here shown, more receptors are found in the soluble fraction of the membrane, suggesting a loss of raft association. And another really important study from 2020 from the Miller lab is that there is a PIP2 binding site on the alpha subunit of GABA-A receptor. However, they did not observe any gating function directly linked to this PIP2 binding site. So taken together, all of this suggests that GABA-A receptor has an affinity for both cholesterol-rich lipid rafts and the PIP2 domains. And furthermore, the integrity of the lipid rafts appear to influence the receptor's localization within the membrane, serving as a structural basis for its potential translocation during functional processes. So with this understanding of two significantly different domains on the membrane for the localization of GABA receptor, we want to know where is the receptor on the membrane, and does it actually have this movement with these two different domains, and whether these domains or the local lipid environment could affect this movement, and what's the functional implication for the mobility. So first of all, we want to know where the receptors are. And to answer this, we need to precisely visualize these domains and determine how the receptor interact with them. And however, these domains I talked about are exceptionally small. They're typically below 200 nanometer in size, making them difficult to resolve with traditional light microscopy. And this is when the dSTORM coming to mind. So dSTORM is a super resolution technique that provides the necessary resolution for our experiment. It could go as low as 20 nanometer, so it's absolutely adequate to study this submicroscopic structures. So dSTORM uses conventional fluorophores as photo switches to visualize cellular structures. And for our experiment, the cells were first cultured in imaging chamber, and to probe the localization of specific lipids and protein, the cells were fixed and labeled with the antibody of fluorescent proteins or fluorescent proteins. And the association between the two probes were then quantified using cross-pair correlation analysis, which generate a pair correlation curve shown here, with the x-axis being the distance between two probes, and the y-axis being the likelihood of their co-localization at the distance. So to analyze the pair correlation data, we took two complementary approaches, which will be the data significance I show later on. So first, we would perform a nested t-test to analyze changes along the broad curve, which provides a detailed picture of how the association changes over distance. And secondly, we quantify the pair correlation at a short radius to focus on local colocalization, presenting the results as bar graphs for better visualization. So for labeling lipid rafts, I use chlorotoxin B, which binds to the GM1 lipids as a well-established hallmark for lipid rafts. And to ensure that GM1 labels accuracy of the lipid rafts, we first validated the labeling with the help of a previous postdoc. So flotilin is a known raft marker, which was used here as a positive control, and the transferrin receptor, which is excluded from rafts from its chemical property, is served as a non-raft control. And here we can see that the GM1 exhibits a high pair correlation with the raft marker flotilin, and a low pair correlation with the non-raft transferrin receptor. And this result supports that GM1 labeling is representative of the lipid rafts. And to simplify the discussion moving forward, I will use the term lipid rafts and the GM1 domains interchangeably. So next we check the localization of GABA-A receptor at a resting state. So here our data shows that GABA-A receptor exhibits a high pair correlation with GM1, supporting the idea that palmitoylation do recruit the GABA-A receptor into the lipid rafts. And this indicates that the resting conformation of GABA-A receptor favors the raft environment. So with this understanding that GABA-A receptor resides within the lipid rafts at a resting state, we want to know, is this localization to the lipid raft static, or does it move on the membrane in response to changes in its activation state? Therefore, I used GABA, the receptor's endogenous agonist, to mimic physiological environment or conditions in activating the receptor. So by comparing the receptor's localization before and after activation, I am to understand how its interaction with lipid domain changes during the functional cycle.